Hello and welcome to another deep dive. Today we're going to unlock a truly fundamental concept in biology. It's absolutely crucial for your class 11th and 12th board exams and of course a high-yield topic for NEET and other state-level medical entrance exams. Respiration in plants. On the surface it might sound simple, plants just breathe, right? But I know there's so much more intricate detail, so much biological genius hidden within this process. I'm really curious to understand how plants, without anything like our lungs or gills, manage this incredibly essential life process. How do they get the energy to grow, to absorb water, to bloom? Can you help us truly unpack this?
Absolutely. That's an excellent question, and it beautifully sets the stage for our deep dive today into respiration in plants. Our mission isn't just memorizing facts, you see. It's about understanding the core principles, why all living organisms, from the tiniest microbes to the largest trees, need a constant supply of energy, and how they meticulously extract this energy from food, and specifically, the sophisticated cellular mechanisms plants employ to do just that. We'll meticulously break down complex cycles like glycolysis, the Krebs cycle, and the electron transport system. We'll focus on every detail you need to master for your competitive exams. By the end of this session, you'll have a crystal clear, structured understanding of how plants truly breathe and derive every bit of their vital energy. So let's take this step by step, building our knowledge foundation. First, let's establish the bedrock of our understanding. Why is respiration so utterly essential for life itself? Think about it for a moment. Every single activity a living organism performs, no matter how small, demands energy. Whether it's the roots absorbing nutrients from the soil, the plant transporting water and minerals all the way up to its highest leaves, the subtle movements within cells, the critical process of reproduction to ensure the next generation, or even our own unconscious act of breathing, all these require a continuous expenditure of energy. So the fundamental question arises, where does all this energy come from? We know that for us, humans and animals, we obtain energy by consuming food. But how is this energy actually liberated from the food we consume? And once released, how is it then efficiently utilized by the cells? Do all types of food, say carbohydrates versus fats, provide the same amount of energy in the same way? And what about plants? Do plants eat food in the way we do, or do they have a different source for their energy requirements? And for microorganisms, are their energy needs met through a similar process of food consumption? These are the foundational questions that respiration answers.
I can see how these seemingly disparate questions actually converge on a central, unifying theme of energy. It sounds like the process of breathing, which we often take for granted, is deeply connected to how all life fuels itself.
Precisely. Your observation is spot on. These questions, while appearing disconnected at first, are indeed profoundly interconnected. The visible process of breathing in macroscopic organisms like ourselves is intimately linked to the microscopic, fundamental process of energy release from food within every single cell. All of the energy required for these life processes is primarily obtained by the oxidation of certain complex organic macromolecules that we collectively term food. Now, for green plants and also certain fascinating organisms like cyanobacteria, they possess a truly remarkable and unique ability. They can prepare their own food. This incredible feat is achieved through the process of photosynthesis, where they effectively trap light energy from the sun and ingeniously convert it into chemical energy. This chemical energy is then stored in the high-energy chemical bonds of carbohydrates, such as glucose, sucrose, and starch. But here's where it gets really interesting, and this is a key conceptual point for your exams. Even in green plants, we must remember that not all cells, tissues, and organs are photosynthetic. Only those cells that contain the specialized organelles called chloroplasts, which are most often located in the superficial or outer layers of the plant, like the leaves, can carry out photosynthesis.
That's a great point. So if only specific cells, mainly in the leaves, are doing all the photosynthesis, how do parts like the roots, which are buried underground, or the woody, non-green parts of the stem, get their energy? Does that mean the food has to be transported to them, much like our blood circulates nutrients throughout our body?
That's an excellent, insightful question, and you've hit upon a crucial mechanism in plant physiology. You're absolutely correct. The carbohydrates, which are the food molecules produced in the photosynthetic cells, must be efficiently translocated or transported to all the non-green parts of the plant for their own energy requirements. This happens through a process called phloem transport. Imagine a root cell working hard to absorb water and minerals deep underground, or a cell in the inner woody part of a tree stem providing structural support. These cells don't have chloroplasts, so they're entirely dependent on the food manufactured in the leaves. They are like, non-producing departments relying on the central factory. And if we zoom out to the broader ecosystem, it becomes even clearer. Ultimately, whether it's animals obtaining food from plants directly as herbivores or indirectly as carnivores, or even saprophytes like fungi and bacteria, depending on dead and decaying organic matter for their sustenance, all the food that is ultimately respired for all life processes fundamentally traces its origin back to photosynthesis. Photosynthesis, as you know, takes place within the chloroplasts and eukaryotic organisms where light energy is transformed into chemical energy. But today, our precise focus is cellular respiration, the specific step-by-step mechanism of breakdown of these stored food materials within the cell to release that trapped chemical energy. And crucially, we'll examine how this released energy is not just dissipated, but intelligently trapped and stored in the form of ATP adenosine triphosphate, which is universally recognized as the energy currency of the cell. This raises an important question for our deeper understanding. When complex compounds like glucose break down during respiration, how exactly is this energy released? Is it all at once in one massive explosive burst?
My intuition tells me it can't be all at once. If it were, wouldn't it be like a huge uncontrolled explosion, releasing so much heat that it would damage or even incinerate the cell? Is that why the process must happen in a series of smaller, more controlled steps?
You're spot on. Your intuition is perfectly aligned with cellular reality. If all the energy contained within a respiratory substrate, say, a glucose molecule were released in a single, uncontrolled step, it would indeed be largely wasted as unusable heat, which would be catastrophic for the delicate machinery of the cell. Think of it like trying to light a matchstick with a flamethrower. It's just too much energy, too fast.
Oh.
So the breaking of the carbon bonds of complex organic compounds through oxidation within the cells, leading to a considerable amount of energy release, is precisely what we define as respiration. The specific compounds that are oxidized during this incredibly vital process are known as respiratory substrates. Usually, carbohydrates like glucose are the primary and most readily available substrates used to release energy. However, it's important for your competitive exams to remember that under certain conditions, or in specific plants or organisms, other macromolecules like proteins, fats, and even organic acids can also be effectively utilized as respiratory substances. Now, what's truly fascinating and a key point for your conceptual understanding is that this energy is not released freely into the cell in a single explosive step. Instead, it is released in a meticulously controlled, slow, and stepwise series of reactions. Each one of these reactions is precisely regulated and catalyzed by specific enzymes. This controlled sequential release is absolutely vital because it allows the energy to be efficiently trapped and conserved as chemical energy in the form of those ATP molecules we discussed. It's critical to remember that the energy liberated by the oxidation and respiration is not used directly by the cell for its activities. Instead, it's like earning money. The energy is used to synthesize ATP molecules. Then, these ATP molecules are like the cash that is broken down whenever and wherever energy needs to be utilized within the organism for absorption, transport, movement, anything. Therefore, ATP effectively functions as the universal energy currency of the cell, much like the Indian rupee is our daily currency for transactions. This sophisticated system ensures highly efficient energy utilization and prevents wasteful heat loss. Beyond just energy, it's worth noting that the carbon skeletons, the intermediate molecules produced during respiration, are also crucial. They are often used as precursors for the biosynthesis of other important organic molecules in the cell, highlighting the interconnectedness of cellular metabolism. #1, do plants breathe? Understanding the basics. Now, let's address a fundamental question that frequently appears in your board exams and NEET. Do plants actually breathe? The answer, as we hinted earlier, is a nuanced but definitive yes. Plants do indeed require oxygen for the process of respiration to occur efficiently, and just like us, they also release carbon dioxide as a metabolic byproduct. However, here's the crucial difference and a common point of confusion for students. Unlike complex animals, plants do not possess specialized respiratory organs such as lungs, gills, or even a sophisticated circulatory system for gas transport. Instead, they rely on much simpler yet highly effective structures for gas exchange with their environment. These are the tiny pores called stomata, primarily found on the surface of leaves, and the small, raised pores called lenticels, which are present on the surface of woody stems and roots. This brings us to a very important question. How do plants manage to respire effectively without these complex organs, especially considering their often immense size and complex architecture?
That's always puzzled me. When I think of a massive tree, like an old banyan, how can it possibly get enough oxygen to its deepest cells without a circulatory system for gases, like we have our blood? It seems like it'd be a huge logistical challenge, almost impossible.
Excellent doubt. That's a very common and insightful question for students preparing for NEET, and understanding the answer will solidify your conceptual clarity. There are actually 3 key reasons why plants are remarkably efficient in their gas exchange despite lacking specialized organs, and these are points you should certainly commit to memory for your exams. First, each plant part largely takes care of its own gas exchange needs. Unlike animals, where a centralized respiratory system, like our lungs, supplies oxygen to virtually all parts of the body via the bloodstream. In plants, there's very little interpart transport of gases. This is a critical distinction. So, a root cell, deep underground, actively obtains its oxygen directly from the tiny air spaces in the soil. A leaf cell, exposed to the atmosphere, facilitates gas exchange through its stomata. And a stem cell, particularly in woody plants, gets its oxygen through the lenticels. Each part operates quite autonomously in terms of its immediate gas exchange requirements. Second, plants in general present a far lower demand for gas exchange compared to animals. Consider the metabolic rates. Roots, stems, and leaves typically respire at rates significantly lower than those of animals. Animals are generally highly active, constantly moving, maintaining a stable body temperature, and engaging in complex physiological processes, all of which lead to a very high and continuous oxygen demand. Plants, on the other hand, are largely sessile. They don't move around, and thus their overall metabolic rate is much slower. The only time plants exchange large volumes of gases is, ironically, during photosynthesis, when leaves actively take in large amounts of carbon dioxide and release oxygen. And critically, during photosynthesis, the availability of oxygen is not a problem in those photosynthetic cells, because oxygen is being produced right there within the cell as a direct byproduct of photosynthesis itself. So the parts that are most active in gas exchange, the leaves, are also the ones producing their own oxygen supply. Third, the distances that gases must diffuse even in seemingly large, bulky plants are surprisingly not great for living cells. You might envision a massive tree and wonder how oxygen reaches its core, but let's clarify. Most living cells in a plant are located quite close to the surface.
Right, that makes sense for leaves, they're thin. But what about thick stems and roots?
Good point. In thick stems, the actively living cells are primarily organized in thin layers just inside and beneath the bark, forming the vascular cambium, phloem, and outer xylem. These layers are directly exposed to the atmosphere via openings called lenticels, which efficiently facilitate gas exchange. The cells in the very interior, the bulk of a thick stem or root, are often composed of dead cells like the heartroot of a tree, which primarily provide mechanical support and do not require oxygen for metabolic activity. This clever structural arrangement, combined with the loose packing of parenchyma cells, which are found throughout leaves, stems, and roots, creates an interconnected network of internal air spaces throughout the plant body. This ensures that the vast majority of living cells in a plant have at least a part of their surface in direct contact with air, allowing for surprisingly efficient diffusion of gases over short distances. So when we encapsulate the entire process, the complete oxidation or combustion of glucose, which is the primary respiratory substrate, ultimately yields carbon dioxide, CO2, and water, H2O, as the final inorganic end products, releasing a significant amount of biological energy. The general chemical equation that summarizes this overall reaction is a cornerstone for your understanding and often appears in exams. C6H12O6 glucose plus 6O2 oxygen is 6CO2 carbon dioxide plus 6H2O water plus energy. This equation is absolutely vital. You should definitely know this one. The plant strategy, and indeed the strategy of all living cells, is to catabolize, meaning to break down the glucose molecule not in one large explosive step, but in a precisely controlled series of many small sequential steps. This multi-step breakdown is the genius of cellular respiration. It allows the energy released at each tiny step to be efficiently captured and coupled to the synthesis of ATP, preventing the majority of it from being lost as unusable heat. This controlled release, this careful dismantling of the glucose molecule to extract its energy, is, in essence, the intricate story of respiration. It ensures that the precious energy is harnessed for cellular activities rather than wasted. During this overall process, oxygen is consumed, and carbon dioxide, water, and energy are the primary products.
So if oxygen is so essential for this complete combustion of glucose, what happens if it's simply not available? Are there organisms or specific situations where respiration has to occur without oxygen? Does the whole energy production just grind to a halt then?
That's a very insightful query, and it leads us to a crucial distinction in the world of cellular respiration. No, energy production doesn't necessarily grind to a complete halt. Instead, the cell employs a brilliant workaround. Yes, absolutely. There are indeed situations and many organisms that not only survive, but actually thrive in environments completely devoid of oxygen, or in very low oxygen conditions. Think about the very early history of life on Earth. The atmosphere billions of years ago largely lacked free oxygen. The first cells on this planet were, by necessity, anaerobic. Even among present-day living organisms, we encounter several fascinating examples adapted to anaerobic conditions. Some organisms are what we call facultative anaerobes, a term you should note down. This means they are versatile. They can switch between aerobic respiration when oxygen is present and anaerobic respiration when oxygen is absent or scarce, depending on the environmental conditions. Others are obligate anaerobes, meaning they are strictly required to live in anaerobic conditions, as oxygen is actually toxic or lethal to them. What's truly fascinating here, and a key conceptual point often tested in NEET, is that all living organisms, regardless of their oxygen requirement, have retained the fundamental enzymatic machinery to partially oxidize glucose without the help of oxygen. This initial breakdown of glucose to pyruvic acid, a pathway common to both aerobic and anaerobic processes, is universally called glycolysis. It is indeed the very first and most ancient step occurring in all organisms, a testament to its evolutionary significance. All right, let's now deeply unpack glycolysis, a pathway you absolutely must master for your NEET and board exams. It's often referred to as the EMP pathway, a mnemonic you'll encounter frequently, named after the pioneering scientists Gustav Emden, Otto Meyerhoff, and J. Parnas, who painstakingly elucidated its intricate scheme. The name glycolysis itself is derived from Greek words, glykos meaning sugar and lysis meaning splitting. So conceptually, it means the splitting of sugar. It is genuinely a universal pathway, occurring ubiquitously in the cytoplasm of every single living organism, from the smallest bacteria to the largest plants and animals. In many anaerobic organisms, glycolysis isn't just a part of respiration. It's the only process in respiration that generates a net gain of ATP. Let's take this step by step, visualizing each stage for your exam perspective. Here's the core transformation that happens in glycolysis. A 6-carbon glucose molecule undergoes a partial oxidation, resulting in the formation of 2 molecules of a 3-carbon compound called pyruvic acid. Now, in plants, this glucose molecule that enters glycolysis is often directly derived from sucrose, which is the primary disaccharide end product of photosynthesis. Alternatively, it can come from stored complex carbohydrates like starch, which are first broken down into glucose. Before glucose can even begin glycolysis, sucrose must first be converted into its constituent monosaccharides, glucose and fructose. This critical conversion is catalyzed by the enzyme invertase. Both glucose and fructose can then readily enter the glycolytic pathway. The very first commitment step, a crucial phosphorylation event, is where glucose and fructose are primed for breakdown. Glucose is converted to glucose 6-phosphate through the activity of the enzyme hexokinase, utilizing 1 molecule of ATP. Similarly, fructose is also converted to fructose 6-phosphate. From this point onwards, the subsequent steps of metabolism for both glucose and fructose are identical, funneling into the same pathway.
So, Professor, if I'm picturing this correctly, glycolysis is a chain of 10 distinct steps, each one precisely controlled by a specific enzyme. And you mentioned ATP being utilized and produced right from the start. So it's a bit of both, an energy investment phase followed by an energy payoff phase, isn't it?
That's a very important insight for your NEET preparation, yes. You've perfectly grasped the dual nature of glycolysis. It is indeed a chain of 10 distinct sequential reactions, each meticulously controlled by a specific enzyme, all taking place in the cytoplasm to transform 1 molecule of glucose into 2 molecules of pyruvate. When you study the detailed steps of glycolysis, it's absolutely crucial to pay close attention to the specific steps where ATP is utilized, meaning consumed or invested, and where synthesized, meaning produced or gained. Also, mark the points where the reduced coenzyme NADH plus H plus is formed, as this molecule carries significant energy to be harvested later. Let's break down the energy changes in detail. First, ATP utilization, the energy investment phase. This initial phase, as you rightly pointed out, actually consumes ATP. Think of it as investing a small amount of capital to get a much larger return later. This energy input primes the glucose molecule, making it unstable and ready for splitting. First, 1 molecule of ATP is used in the very first step, the conversion of glucose into glucose 6-phosphate. This phosphorylation makes glucose more reactive and traps it inside the cell. The enzyme for this is hexokinase. 2. Second, another molecule of ATP is utilized later on when fructose 6-phosphate is converted to fructose 1,6-bisphosphate. This step adds a second phosphate group, further destabilizing the sugar molecule, preparing it for cleavage. So in total, 2 ATP molecules are invested or consumed in these initial priming steps. Now, energy payoff phase and NADH plus H plus formation. After this initial investment, the pathway begins to yield energy. This is where the cell starts to reap the benefits. 1. The 6-carbon fructose 1-valent 6-bisphosphate molecule then performs a remarkable split into 2 distinct 3-carbon compounds, dihydroxyacetone phosphate, DHAP, and 3-phosphoglyceraldehyde, PGL. Interestingly, DHAP is readily and reversibly converted into PGL. So for all practical purposes and for energy calculations, one glucose molecule effectively yields 2 molecules of PGL, which then proceed through the rest of the pathway. 2. Here's where NADH plus H plus is formed, a crucial redox reaction for capturing high-energy electrons. Each molecule of PGAL undergoes oxidation, and with the simultaneous incorporation of inorganic phosphate, it gets converted to 143-bisphosphoglycerate, BPGA. During this oxidation, 2 redox equivalents, which are essentially packages of high-energy electrons and protons, are removed from PGL and immediately transferred to a molecule of NAD plus pi equals CO, forming NADH plus APLH plus L-FO.
Okay, so since we have 2 PGLs from glucose, we get 2 NADH plus H plus molecules here.
Exactly. 2 molecules of NADH plus H plus red. These NADH molecules are like energy vouchers, holding potential energy. 3. Now, for ATP synthesis, which occurs via a mechanism called substrate-level phosphorylation, the conversion of BPGA, 1,3-bisphosphoglycerate, to 3-phosphoglyceric acid, PGA, is an energy-yielding process. The energy released from the breaking of a high-energy phosphate bond within the substrate itself is directly utilized to form ATP from ADP and inorganic phosphate. This is like the cell's quick cache ATM, where energy is transferred directly without a complex chain. Since we have 2 BPGA molecules, 2 ATPs are formed at this specific step. 4. Another ATP is synthesized during the final energy-yielding step, the conversion of phosphenyl pyruvate, PEP, to pyruvic acid. Again, this is another crucial instance of substrate-level phosphorylation, and since there are 2 molecules of PP, 2 molecules of ATP are formed here. So if you meticulously calculate the net direct gain of ATP from glycolysis for every glucose molecule that enters the pathway, here's what we get. We invested 2 ATPs initially, but we produced 4 ATPs directly through substrate-level phosphorylation, 2 from BPGA to PGA, and another 2 from PP to pyruvate. Therefore, the net direct gain of ATP from glycolysis is 4 produced, 2 utilized, equals 2 ATP molecules. This is your net profit from glycolysis. Pyruvic acid, a 3-carbon compound, is the key final product of glycolysis. Now, this is a critical branching point. The metabolic fate of pyruvic acid is not fixed. It is entirely dependent on 2 crucial factors: the cellular needs of the organism and, most importantly, the availability of oxygen in the cell's environment. This leads us directly to the next stage of respiration. What happens to pyruvate next? From pyruvic acid, there are 3 major metabolic pathways it can take, forming a crucial decision point in cellular energy metabolism depending on the conditions within the cell. These are lactic acid fermentation, alcoholic fermentation, and aerobic respiration. Today, we'll first focus on fermentation, which occurs specifically under anaerobic conditions, meaning in the complete absence of oxygen. This pathway is a critical survival mechanism common in many prokaryotes, such as certain types of bacteria, and importantly, in unicellular eukaryotes like yeast. It's a vital alternative for organisms when oxygen is scarce. Let's delve into the 2 primary types of fermentation relevant for your exams.
So if I'm understanding this correctly, fermentation is like the cell's crucial plan B, or backup strategy. When oxygen, the preferred fuel for maximum energy, isn't around, fermentation is a way for the cell to keep generating at least some energy, even if it's not ideal, just to survive and keep glycolysis going.
Precisely. You've captured the essence perfectly. It's indeed the cell's crucial strategy to generate a limited amount of ATP and, even more importantly, to regenerate the crucial coenzyme NAD+ base. This allows glycolysis, the initial energy-producing step, to continue unhindered, even in the absence of oxygen. Without NAD+ base, glycolysis would simply stop. Let's look at the 2 main types. 1. Alcoholic fermentation. This pathway is prominently observed in yeast, the same yeast used in baking and brewing. In alcoholic fermentation, pyruvic acid is converted into 2 final products: carbon dioxide, CO2, and ethanol, alcohol. This process involves 2 key enzymatic steps. First, the enzyme pyruvic acid decarboxylase removes a molecule of CO2 from pyruvic acid, producing a 2-carbon compound called acetaldehyde. Second, the enzyme alcohol dehydrogenase then reduces this acetaldehyde to ethanol, utilizing the NADH plus H plus that was produced during glycolysis. This step regenerates NAD plus pyruvate, lactic acid fermentation. This pathway is common in some bacteria, such as lactobacillus species used in making yogurt and cheese, and notably, it also occurs in our own muscle cells during periods of intense physical exercise.
Ah, like when you sprint and your muscles start to burn. That's lactic acid buildup.
Exactly that. Imagine a strenuous sprint where your oxygen supply to muscles can't keep up with the high energy demand. In this scenario, pyruvic acid is directly reduced to lactic acid. This reaction is catalyzed by the enzyme lactate dehydrogenase, which, like alcohol dehydrogenase, also utilizes NADH plus H plus and regenerates NAD plus Phet. This regeneration of NAD+ is vital for glycolysis to continue. In both of these fermentation processes, alcoholic and lactic acid fermentation, a critical and common step occurs. The reducing agent, NADH plus H+, which was formed during the oxidation step of glycolysis, is oxidized back to NAD plus Phet. This continuous regeneration of NAD+ is absolutely crucial for glycolysis to proceed. Without a constant supply of NAD+, glycolysis would grind to a complete halt, thereby stopping even the limited ATP production that fermentation offers. So while it may seem like a simple side reaction, this NAD+ regeneration is fundamental for sustaining energy production under anaerobic conditions. Now, what's important for your competitive exams, and a major point of contrast, is to note the energy yield here. Fermentation releases a very small fraction of the total energy contained within the glucose molecule. Typically, less than 7% of glucose's inherent chemical energy is actually released. And not all of that small amount is trapped as usable ATP. You only get a net gain of 2 ATPs, which are the 2 ATPs directly produced from glycolysis itself. This is remarkably inefficient compared to aerobic respiration, which we'll discuss next. Furthermore, the products of fermentation, either acid, like lactic acid, or alcohol, like ethanol, can become hazardous or even toxic to the organism if they accumulate in high concentrations. Yeasts, for example, which are fascinating microorganisms, literally poison themselves to death when the concentration of alcohol they produce reaches about 13%.
Wow, really? So they basically die from their own waste product.
That's right. This biological limit explains why naturally fermented beverages, such as wine or beer, typically have a maximum alcohol content around that percentage. To achieve higher alcohol concentrations, like those found in spirits such as whiskey or vodka, distillation, a process of separating alcohol from the water and other compounds, is required. This inherent inefficiency and potential toxicity highlight why organisms, whenever possible, overwhelmingly prefer the aerobic pathway for energy extraction.
So if fermentation is so inefficient, barely yielding any ATP, and even produces toxic byproducts, then aerobic respiration must be where the real energy gain happens. It sounds like it's the cellular equivalent of a massive, highly efficient power plant compared to a small, inefficient hand generator. How does the cell extract a much, much larger amount of energy with oxygen, making it so dramatically more effective?
That's an excellent analogy. You've perfectly captured the profound difference. This is indeed where the true energy powerhouse of the cell comes into play, generating the vast majority of ATP for most higher organisms. In eukaryotic cells, this incredibly intricate and efficient process predominantly takes place within the mitochondria, which are famously referred to as the powerhouses of the cell. Aerobic respiration is defined as the complete oxidation of organic substances, primarily glucose, which occurs crucially in the presence of oxygen. This pathway meticulously breaks down glucose, releasing a massive amount of energy along with carbon dioxide, CO2, and water, H2O, as the final harmless inorganic products. This is the most common and unequivocally the most efficient type of respiration found in higher organisms, including plants. For aerobic respiration to proceed, the final product of glycolysis, pyruvate, a 3-carbon compound that was formed in the cytoplasm, must first be transported from the cytoplasm into the mitochondrial matrix. This transport is itself a regulated process. Once inside the mitochondria, 2 crucial sequential events define the core of aerobic respiration. Crucial event 1, complete oxidation of pyruvate. This involves the stepwise and meticulous removal of all hydrogen atoms from pyruvate. Each carbon atom is ultimately released as CO2. This primarily occurs within the mitochondrial matrix and encompasses a preparatory step, the linking reaction, followed by the Krebs cycle. Crucial event 2, ATP synthesis through electron transfer. This is where the bulk of the energy is harvested. The electrons, which were removed as part of these hydrogen atoms during the oxidation of pyruvate, are then passed efficiently to molecular oxygen, O2. Oxygen acts as the final electron acceptor. This highly controlled electron transfer is precisely coupled with the simultaneous synthesis of a very large amount of ATP. This second process is located on the highly folded inner mitochondrial membrane. Let's first follow pyruvate as it makes its way into the mitochondria. Pyruvate, as you recall, is a 3-carbon compound formed from the glycolytic breakdown of carbohydrates in the cytosol. Once it crosses the outer and inner mitochondrial membranes and enters the mitochondrial matrix, it undergoes a pivotal transformation called oxidative decarboxylation. Think of this as a crucial linking reaction that connects glycolysis to the main power-generating cycles. This is a complex set of reactions catalyzed by a large multi-enzyme complex known as pyruvic dehydrogenase. This complex requires the participation of several crucial coenzymes, including NAD+ and coenzyme A, so CoA, as well as magnesium ions, Mg2+. The overall reaction can be precisely summarized as pyruvic acid plus CoA plus NAD plus acetyl-CoA plus CO2 plus NADH plus H plus 8. Remember, the enzyme complex and Mg2+ are needed.
Okay, so pyruvate loses a carbon as CO2 and becomes acetyl-CoA, which is a 2-carbon molecule, and we get NADH2.
Exactly. This is a critical step for your competitive exams. A carbon atom is released as CO2, and energy in the form of NADH plus H+ is captured. Remember, since one glucose molecule yields 2 molecules of pyruvic acid during glycolysis, this oxidative decarboxylation step per glucose molecule effectively produces 2 molecules of acetyl-CoA, 2 molecules of CO2, and 2 molecules of NADH plus H plus 1. This acetyl-CoA molecule is now primed and ready to enter the core of aerobic respiration, the Krebs cycle, hashtag 4.1, the tricarboxylic acid cycle, TCA cycle, or Krebs cycle. From acetyl-CoA, we now enter a remarkable cyclic pathway, famously known as the tricarboxylic acid, TCA cycle, or more commonly, the Krebs cycle. It's named after the Nobel laureate Sir Hans Krebs, who meticulously elucidated the central metabolic pathway. This cycle is absolutely central to aerobic respiration, as its primary function is the complete oxidation of the 2-carbon acetyl group derived from pyruvate. Let's visualize this cycle step by step for your complete understanding, as it's a frequently tested topic. Step 1, condensation. The Krebs cycle initiates with a highly significant condensation reaction. The 2-carbon acetyl group from acetyl-CoA combines, with a 4-carbon molecule called oxaloacetic acid, OAA, in the presence of water. This reaction yields a 6-carbon compound, which is citric acid, hence why this cycle is also known as the citric acid cycle. This crucial initial reaction is catalyzed by the enzyme citrate synthase, and importantly, coenzyme A, CoA, is simultaneously released, allowing it to be recycled. Step 2, isomerization. The 6-carbon citrate molecule then undergoes an isomerization reaction. This means its atoms are rearranged to form an isomer called isocitrate. This step simply changes the molecular structure, preparing it for the next stages. Step 3 and 4, decarboxylations and NADH formation. Next, we encounter 2 successive and extremely vital steps of decarboxylation. Decarboxylation means the removal of a carboxyl group, which is released as a molecule of CO2. These steps are crucial for releasing carbon atoms with carbon dioxide and capturing energy in the form of reduced coenzymes. First, isocitrate is oxidized and decarboxylated to form iketoglutaric acid, which is a 5-carbon compound. During this step, 1 molecule of NAD+ is reduced to NADH plus H plus 1. Second, iketoglutaric acid is also oxidized and decarboxylated to form succinyl-CoA, a 4-carbon compound. Again, another molecule of NAD+ is reduced to NADH plus H plus O.
So these 2 steps release the remaining 2 carbons from the original acetyl-CoA as CO2.
Precisely. The 2 carbons from acetyl-CoA are released as CO2 in these decarboxylation steps, along with the 1 CO2 released during the linking reaction. That accounts for all 3 carbons from the original pyruvate. Step 5, substrate-level phosphorylation. This is a direct energy-yielding step within the Krebs cycle. During the conversion of succinyl-CoA to succinic acid, another 4-carbon compound, a molecule of GTP, guanosine triphosphate, is synthesized directly from GDP, an inorganic phosphate. This is a crucial example of substrate-level phosphorylation within the Krebs cycle, similar to what we saw in glycolysis. This GTP is then readily converted to GDP with the simultaneous synthesis of ATP from ADP. So effectively, 1 ATP molecule is directly produced here for each turn of the cycle. Remaining steps. Regeneration of OAA. The remaining steps of the cycle involve a series of oxidation reactions that meticulously convert succinic acid back to oxaloacetic acid, OAA, which is the starting molecule. This regeneration of OAA is absolutely critical because it ensures the cycle can continue to accept new acetyl-CoA molecules. During these final transformations, we see further reductions. There are 2 more points in the cycle where NAD+ is reduced to NADH plus H plus ec, and more significantly, 1 point where F80+ is reduced to FADH2 during the conversion of succinate to fumarate. So let's tally the energy-rich molecules produced for each acetyl-CoA molecule that enters the Krebs cycle. You get 3 molecules of NADH plus H plus A, 1 molecule of FADH2, and 1 molecule of ATP or GTP. Since one glucose molecule yields 2 acetyl-CoA molecules, these numbers effectively double per glucose molecule that started the whole process. So for one glucose molecule, the Krebs cycle, after processing 2 acetyl-CoA, yields a total of 6 NADH plus 80 plus USSN, 2 FADH2, and 2 ATP. The cycle also critically requires the continuous replenishment of oxaloacetic acid to serve as the initial acceptor for acetyl-CoA, and the continuous regeneration of NAD plus and FAD plus for it to keep running efficiently.
Okay, Professor, so we've meticulously broken down glucose completely to CO2 by the end of glycolysis in the Krebs cycle. And we've generated a lot of those high-energy electron carriers, NADH and FADH2, plus a little ATP directly. But I still haven't seen much oxygen being utilized directly in these steps, nor the large number of ATP molecules that was promised for aerobic respiration. What's the real role of all this NADH and FADH2, and where exactly does oxygen come in to yield that huge energy boost we talked about? This seems like the final critical piece of the puzzle. Hashtag tag 4.2. Electron transport system, ETS, and oxidative phosphorylation.
Excellent question. You've precisely identified the grand finale, the stage where the bulk of ATP is generated and where oxygen plays its absolutely vital, indispensable role. This is the electron transport system, ETS, also known as the electron transport chain, and it's the metabolic pathway through which the energy meticulously stored in NADH plus H plus and FADH2 is finally harnessed. In this system, electrons are systematically passed from one carrier molecule to another, moving like a relay race, ultimately reaching oxygen. This final electron transfer to oxygen results in the formation of water. Crucially, this entire electron transport process is tightly coupled with a massive burst of ATP synthesis. This intricate system is located on the highly folded inner mitochondrial membrane, providing a vast surface area. Let's take this step by step, focusing on the specific complexes, as understanding their roles is paramount for your exams. Picture this. Imagine a series of highly organized protein complexes, much like specialized machinery, embedded within the inner mitochondrial membrane. These complexes act as a precise relay team for electrons. Complex I, NADH dehydrogenase. This is the first entry point for electrons from NADH. Electrons from NADH plus H+, producing glycolysis and Krebs cycle, are oxidized here. This complex then transfers these electrons to a mobile electron carrier called ubiquinone, complex 2 succinate dehydrogenase. This complex acts as a separate entry point. Ubiquinone also receives reducing equivalents, electrons, directly from FADH2, which was generated during succinate oxidation in the Krebs cycle. Complex 2 facilitates this transfer directly to ubiquinone.
So FADH2 electrons skip complex I.
Yeah. That's right. They enter at complex 2, feeding into ubiquinone, bypassing complex 1. This is important for the ATP yield later. Complex 3, cytochrome BC1 complex. The reduced ubiquinol, then transfers its electrons to cytochrome C. Cytochromes are specialized proteins containing iron ideal electron carriers. Cytochrome C, this is a small mobile protein on the outer surface of the inner membrane acting as a carrier between complex 3 and 4. Complex 4 cytochrome C oxidase complex, this is the terminal complex. It contains cytochromes A and A3 and 2 copper centers. This complex is responsible for the final critical transfer of electrons to oxygen. Now here's the brilliance of the system. As electrons move sequentially from one carrier to another through these complexes, 1 to 5 or 2 to 3 to 4, they are moving from a higher energy level to a lower energy level. The energy released from this stepwise downhill movement of electrons is not wasted. Crucially, this energy is precisely utilized to pump protons, H+ ions, from the mitochondrial matrix into the intermembrane space. This active pumping creates an electrochemical proton gradient across the inner mitochondrial membrane. This gradient is a form of stored potential energy, like water behind a dam. Protons are concentrated on one side, eager to flow back.
Okay, so the electrons moving power proton pumps, creating a gradient. But what about oxygen? You said it was vital.
Absolutely vital. Oxygen's role, though seemingly limited to the very terminal stage at complex 4, is indispensable. It acts as the final electron and hydrogen acceptor. By accepting the electrons from complex 4 and then combining with protons in the matrix to form water, H2O, oxygen effectively pulls the entire electron transport chain forward. It's like the vacuum cleaner at the end, constantly clearing the pathway. Without oxygen to act as this final acceptor, the electrons would back up, the entire chain would halt, the proton gradient would dissipate, and ATP production would cease. This explains why organisms with high energy demands cannot survive without oxygen. Finally, let's discuss ATP synthase, often referred to as complex V. This remarkable enzyme is precisely positioned to harness the energy from the proton gradient to synthesize ATP. It acts like a tiny molecular turbine. ATP synthase consists of 2 major components, important to distinguish for your exams, F1 and F0. The F1 headpiece is a peripheral membrane protein complex. It protrudes into the mitochondrial matrix and is the actual catalytic site where ATP synthesis occurs, combining ADP and inorganic phosphate pi to form ATP. The F0 component is an integral membrane protein complex embedded within the inner membrane. F0 forms a channel or a rotating motor through which protons, H+, flow back down their electrochemical gradient from the intermembrane space into the mitochondrial matrix.
So the protons flowing back through F0 power the F1 part to make ATP.
Exactly. The passage of these protons through the F0 channel is directly coupled to the catalytic site of the F1 component. This proton flow powers a rotational movement within F0 and F1, which drives conformational changes in F1, forcing the synthesis of ATP. This elegant mechanism is explained by the chemiosmotic hypothesis. For your exam, remember the crucial ATP yield from the reducing equivalent. The oxidation of one molecule of NADH plus H plus through the ETS gives rise to approximately 3 molecules of ATP, while the oxidation of one molecule of FADH2 produces approximately 2 molecules of ATP. This difference is because FADH2 enters at complex 2, bypassing complex 1 and thus pumping fewer protons. Now let's synthesize all this information and construct the theoretical maximum ATP gain from the complete aerobic respiration of one glucose molecule. While it's crucial for your understanding of energy efficiency, it's very important to remember that this calculation is largely a theoretical exercise. These formulas are derived based on certain ideal assumptions that rarely hold perfectly true in a complex dynamic living system. These assumptions include, first, a perfectly sequential, orderly pathway. Glycolysis, then TCA cycle, then ETS, linearly. Second, all NADH from glycolysis is efficiently transferred into the mitochondria for oxidative phosphorylation. This might have a small cost. Third, no intermediates are siphoned off for other biosynthesis pathways. And fourth, only glucose is being respired, no fats or proteins entering midway.
All right, so real life is messier, but this calculation helps understand the potential.
Exactly. In reality, pathways work simultaneously. Substrates withdrawn, ATP is used constantly, and enzymes are regulated. It's a dynamic web. Yet, this theoretical exercise is extremely useful. It allows us to truly appreciate the sheer beauty, precision, and remarkable efficiency of the living system in extracting energy. Based on these theoretical assumptions, the net gain of ATP during the complete aerobic respiration of one glucose molecule is estimated to be about 38 ATP molecules. This is a figure you should know for your exams.
38 ATPs from just one glucose molecule. Professor, that's an absolutely monumental jump from the mere 2 ATPs we get in fermentation. That difference truly highlights why oxygen is so crucial, doesn't it? It's like the difference between generating power with a small battery versus a massive power grid. The efficiency gain is incredible.
Absolutely. It's a truly significant difference in energy yield, and it unequivocally demonstrates the enormous evolutionary advantage of aerobic respiration. Let's quickly compare fermentation and aerobic respiration side by side, as this is a common comparative question in your exams, summarizing the key distinctions. Breakdown extent. Fermentation is partial breakdown. Products like alcohol lactic acid still have energy. Aerobic respiration is complete breakdown to CO2 and H2O. ATP gain. Fermentation yields only a net of 2 ATPs from glycolysis. Aerobic respiration generates much more, theoretically up to 38 ATPs. NADH oxidation. NADH is reoxidized slowly in fermentation, just enough to keep glycolysis going. In aerobic respiration, NADH and FADH2 reoxidation via ETS is very vigorous and rapid, essential for the high ATP yield. Traditionally, respiration has often been conceptualized as a purely catabolic process, meaning it's solely about breaking down complex substrates like glucose to release energy. However, for a more accurate and comprehensive understanding, especially for advanced biology, the respiratory pathway is better and more accurately described as an amphibolic pathway. This raises an important question. What exactly does amphibolic mean, and why is this classification more accurate than simply labeling it as catabolic?
Amphibolic, that sounds like it has a dual role, not just one. Are you saying, Professor, that the respiratory pathway isn't just about breaking things down for energy, but also about building things up? That seems almost contradictory to its core function of energy release. How can it be both destructive and constructive at the same time?
Exactly. You're thinking critically, and that's precisely what amphibolic implies. It's a metabolic crossroads, a two-way street involved in both catabolism, breaking down, and anabolism, building up. While glucose is indeed the favored substrate, other complex organic substrates like fats and proteins can also be respired. When these are used, they enter the pathway at various intermediate points. Let's illustrate with some examples important for understanding metabolic interconnectedness. Fats. Fats break down into glycerol and fatty acids. Fatty acids are degraded to acetyl-CoA, entering Krebs cycle. Glycerol is converted to PGAL, entering glycolysis. Proteins. Proteins break into amino acids. After deamination, removing the amino group, the carbon skeletons can enter as pyruvate, acetyl-CoA, or Krebs cycle intermediates like alpha-ketoglutarate. What's truly fascinating here, and absolutely crucial to understanding the amphibolic nature, is that these very same intermediate compounds can be tapped into and withdrawn from the pathway to be used for the synthesis of other complex molecules.
So the pathway intermediates aren't just stepping stones for breakdown. They can be pulled out to build other things.
Precisely. It's like a highly efficient factory where intermediates from one line become raw materials for another. For example, if the organism needs to synthesize fatty acids, acetyl-CoA would be withdrawn from the respiratory pathway. Similarly, respiratory intermediates like pyruvate and Krebs cycle intermediates are crucial starting points for synthesizing amino acids and thus proteins. Since the respiratory pathway is actively involved in both breakdown, catabolism, and AND synthesis, anabolism, it's correctly termed an amphibolic pathway. This classification highlights the incredible interconnectedness, flexibility, and resourcefulness of metabolic pathways within a cell. Respiration isn't just destructive, it's also fundamentally constructive. Finally, let's discuss another incredibly important concept, especially for your practical examinations, viva voce, and objective questions in competitive exams like NEET, the respiratory quotient, or RQ. The RQ is a quantitative diagnostic tool defined as the ratio of the volume of carbon dioxide, CO2, evolved to the volume of oxygen, O2, consumed during respiration. So the formula is RQ, Ocostall volume of CO2 evolved, volume of O2 consumed. It's a valuable indicator because it provides crucial insights into the type of respiratory substrate that is being predominantly used by an organism. It's like taking a metabolic fingerprint.
So, Professor, different types of food or substrates give different RQ values. It's like each fuel leaves a unique signature in terms of CO2 produced and O2 consumed.
Precisely. You've grasped the core concept beautifully. The RQ value indeed depends significantly on the chemical composition of the substrate. Let's look at the characteristic RQ values essential for your exams. Carbohydrates. When carbohydrates like glucose are completely oxidized, the RQ is exactly 1.0. This is because equal amounts of CO2 are evolved and O2 are consumed. Recall the equation, C6H12O6 plus 6O2, M6CO2 plus 6H2O. So RQ equals 6CO2, 6O2 equals 1.0. Fats. When fats are used, the RQ is consistently less than 1, typically around 0.7. Fats are richer in carbon and hydrogen, poorer in oxygen, so they need more O2 for oxidation relative to the CO2 produced. For example, tripalmitin, 2C51H98O6 plus 145O2 plus 98H2O, RQ equals 102, CO2, 145O2, M0.7. This lower RQ clearly indicates fat respiration. Proteins. For proteins, the RQ value is approximately 0.9. It reflects their unique composition, somewhere between fats and carbohydrates. Organic acids. Interestingly, for some organic acids, the RQ can even be greater than 1. This happens if the acid already contains a lot of oxygen relative to carbon, requiring less external O2. It's important to recognize that in living organisms, substrates are rarely pure. Often, multiple substrates are respired simultaneously. Pure proteins or fats are almost never the sole substrates normally. This understanding of RQ is incredibly useful. For instance, in germinating fatty seeds, RQ would initially be 1, then shift towards 1 as carbohydrates become the fuel. It helps interpret metabolic states. #outro. So to quickly summarize our truly comprehensive deep dive into respiration in plants, covering everything crucial for your exams. First, all life needs energy from food oxidation, trapped as ATP, the energy currency. Second, plants do breathe, using stomata and lenticels, managing efficiently due to localized needs, lower demands, and short diffusion distances via air spaces. Third, glycolysis in cytoplasm is a universal partial breakdown of glucose to pyruvate, with a net direct gain of 2 ATP and 2 NADH. Fourth, under anaerobic conditions, fermentation, lactic acid or alcohol, regenerates NAD+, but yields only 2 ATP net, making it inefficient. Fifth, aerobic respiration in mitochondria, with oxygen, starts with pyruvate forming acetyl-CoA, linking reaction, producing CO2 and NADH. Sixth, acetyl-CoA enters the Krebs cycle, TCA cycle, in the matrix, fully oxidizing the acetyl group, generating NADH, FADH2, and some ATP, a GP. Seventh, the electron transport system, ETS, on the inner mitochondrial membrane uses NADH and FADH2. Electrons flow through complexes, pumping protons. Oxygen is the final acceptor, forming water. This drives ATP synthase via oxidative phosphorylation to make lots of ATP. NADH, 3 ATP, FADH2, ATP. Eighth, The respiratory balance sheet shows a theoretical maximum of 38 ATP per glucose aerobically, far more than fermentation's 2 ATP, highlighting oxygen's importance. Ninth, respiration is an amphibolic pathway involved in both catabolism, breakdown for energy, and anabolism, providing intermediates for synthesis. And finally, 10th, the respiratory quotient RQ, CO2 evolved, O2 consumed, indicates the substrate, 1.0 for carbs, 1.7 for fats, EV 0.9 for proteins.
Wow, Professor, that was a truly comprehensive and incredibly clear deep dive. From understanding why plants don't need lungs, to the intricate dance of electrons, and even how different foods leave a unique metabolic signature. It's clear how essential and profoundly fascinating cellular respiration is. I feel much more confident about tackling those detailed exam questions now. Thank you for making these complex processes so much more intuitive.
That's truly wonderful to hear. Remember, this chapter is absolutely fundamental, not just for exams, but for understanding biological energy. My advice to you and to all students preparing for NEET and your board exams: revise these pathways again, maybe draw them out, focus keenly on the intermediates, the enzymes, and particularly the energy yields ATP, NADH, FADH2 at each stage. Practice past year questions extensively, especially on ATP calculations, RQ values, and the differences between aerobic and anaerobic respiration. Consolidate these concepts. And as always, please do not hesitate to reach out if you have any further doubts on this chapter. Keep learning, keep exploring, and keep up the hard work for your exams.